Welcome you all to this meeting, arranged by Simon O'Connor. Many thanks for that. And we have a very interesting speaker today. We've heard him once before, fairly recently, but things are changing so fast. So he has a lot more to tell us. It's Jorg Hardege from the University of Hull, and he works on marine invertebrates and how olfactory cues are changing because of climate change and the effect it is having on life at this level. His talk is called The Use of Olfactory Cues in a Changing Environment. You're very welcome to ask questions via the chat, but please mute yourselves now for the talk. Jorg, I hand over to you.
Okay, thank you very much for that lovely introduction. Yeah, I mean, it's about a couple of years ago, well, 3 years now that I gave a presentation. And you'll be interested to see that the science is going that quickly in this field that I've changed the talk quite considerably. So this first slide shows you that we mainly work with marine invertebrates. So this is things like mating behavior on those crabs or these worms here, but also a little bit with fish. So there's some vertebrates, not many, where we are mainly marine, but we also work with crayfish. Over the last few years, my team has grown quite dramatically, and we now have about 3 academics, 3 postdocs, just over 20 PhD students, and about 6 master students in the group. So they're producing data absolutely constantly, and I'm struggling to keep up with them, to be brutally honest. Right, let's see what we're doing. How does normally, I only need to click. So here's what the research group is all about. So the research group started really with identifying and purification of chemical signals, because when we started all that research, very few marine cues were known. And I did my PhD and then postdoc in a chemistry lab. So I learned HPLC and GC and mass spectrometry and all these sort of things. So to identify cues, and we carried on with that for quite a few years. But the more cues we knew, the more the interest shifted into trying to understand what these cues actually do. And whether there is a little bit more complexity in those behaviors. So we started with relatively simple things like marine worms that undergo a metamorphosis and therefore really are very easy to work with, because they only have one thing in mind, reproduction, therefore only use sex pheromones and don't respond for food, for example. And then we went to a more complex animals where even social hierarchies play a role. Knowing more and more marine cues, we thought there's probably applications of these ones, and I started a few projects on using chemical signals, pheromones in pest control. And this is a picture of the deep, where we have a big so-called total environment simulator. It's a big flume in which we put our tanks and traps, for example, to see how these cues can be used in the field. But then for the last 10 years, it's shifted a lot more into what effects humans have on the olfactory systems and the animal's ability to actually use them correctly. And we used to initially develop biomarkers for fitness. And then we worked on these lovely things here in the middle, ocean acidification. It's not lovely, but these lovely things are vents. And this is a CO2 vent. I took a photo at the island of Ischia in Italy, where CO2 bubbles into the water and acidifies the water surrounding. It's a bit like a research lab in the field. And you can use that one to look at adaptations, for example, of the animals living there. But descriptive research wasn't really my thing, so we developed the group further, trying to understand why behaviors change, and then why this is happening with those chemicals. You see the 2 pictures already there, which is electron density maps of signaling cues. Right, okay, let's see what happens. I put in blue here the uptake from this slide. It says olfactory signals are potentially vulnerable to disruption at any stage of their use. Okay, so if we look at that, then it's relatively straightforward that cues are made by a sender, or the cues are taken up and modified to be used, and mainly can be interfered with by the fitness of the animal, even as direct as the making of the cues could be stopped. So making a pheromone and a chemical signal to be released is something that can be interfered with. Once the cues are then in the environment, there's lots of things that can go on, and one on the list here is pH. pH, for example, but also cues can potentially link to pollutants, effective microbes if the cue is there in the environment for a long time, for example. But there's also the main problem, which is the cue needs to reach the receiver. So currents, etc., will all interfere the transmission. Once the cue actually arrives at the receiver, there's a couple of things that have to go right. First is the receptor-ligand interaction, so the chemoreception really, and then later on, it's also obviously the translation of that into behavior. That can be directly affected by affecting the fitness of the animals, or indirectly, for example, by affecting the neurotransmitters that are involved in perception. So in other words, what we look at olfactory cues is quite a vulnerable thing. It can be interfered with at so many different places that it is quite amazing that this works in so many species. So what is my research lab looking at? Well, looking at this problem here. If you look at the graph, it shows you the amount of CO2 in the air. And we all know how much is going up. Actually, this curve is actually going up quicker and quicker and quicker. Parallel in light green here is actually in the ocean, for example, the pH that goes down. So what we get is from now 400, we are expected to have about 800 ppm CO2 by the end of the century. And the latest predictions are for the oceanic pH to drop significantly. So lots of questions arise, and the 4 in blue are the ones where we're actually all involved in by researching that. The first is, what impact does it have? So on behavior, functional traits. Does it mean that animals in a changing environment have their olfactory communication interfered with? And in which way? And how does it work? What actually happens? And then more recently, we're looking at, can animals actually adapt to that? Because on the time scale here, you see that it's a very, very short period of time. And if the animals can adapt, what are the costs to this? Do that at the cost of some fitness? Is it that they have to switch from one sensory mode to another? And are there any tipping points? Is there a sort of level of CO2 or pH that is as far as it goes, and then the animals can't? The last one is anything that can be done, but I will say CCOP 26.54321. I think you can judge on yourself how successful we have been so far as humans to stop that. Right, okay, go one step backwards. I work mainly with marine organisms, and it's relatively straightforward, really. CO2 comes into the air from whatever industrial sources. We can spend the afternoon discussing that. But in principle, all what happens is that the CO2 gets into the water. The oceans are a major sink. You get a chemical reaction. Carbonic acid is formed, and as the result of that one is then an increase in carbonate ions, and what I'm interested in, the increase in hydrogen ions. More hydrogen, yeah, first year chemistry for those who are not chemists. Remember, more hydrogen means negative logarithm, so lower pH. The more hydrogen in the water, the lower the pH is. The more chemicals are in there can be protonated because they pick up the proton. Yeah. Okay, so that's what happens chemically. And what does it mean to the oceanic pH? This is the mean average, or the average pH in open water over the last, 25 million years. Okay, the scale is definitely not in the way that it should be made, but it's there just to show you the short period of time that the major change has happened. So in the last couple of hundred, well, there's over 300 years, we see a drop from about 8.15, 8.2 to 7.7. And then every time there's a new prediction that seems to go lower. Yeah, so we are at 7.6, 7.7 now as a prediction. Doesn't sound much, but remember it's a logarithmic scale. So 1 up means a lot more hydrogen in the water. As a consequence, and that is something interesting that we have started to do for a few years now, is we look at the fluctuations of the pH in the ocean. This is the average, right? But if you look at pH in a coastal environment, you'll see significant differences between night and day. In the night, the pH is much lower than during the day. During the day, we've got algae, microorganisms all producing oxygen and consuming CO2, and in the night it's the opposite way around. So here's calculations that have been published 2 years ago, well, 2 years ago, I know, about how the fluctuations in near coast ecosystems will change. Whilst in pre-industrial times it went up to 8.4 roughly, and went down in the night to about 8. Currently, it's about 8.16 or something like that now, and the low points is already well below 8. So 7.8 or so. And you can see the amplitude here increasing dramatically. In the year 2000, we'll see a major shift. We'll see that even during the day, the pH will struggle to reach 8. And during the night, you will see that is below 7.5. Now, the latest calculations are actually below 7.4, and in areas where there's freshwater intake, it might be even lower. So it is actually correct that animals may be in an environment that is totally unfavorable for them. Equally, one has to think about animals that live and, for example, forage in the night. They will be exposed to much lower pHs than the ones that do the same thing during the day. So what does it mean? It means that many species will have very different effects on their behaviors when these fluctuations really increase. Now, what has been known on olfactory disruption? Well, it started really at the time when we wanted to publish our first papers, was that Danielle Dixon, PhD student of Philip Monday's, published a paper that clownfish larvae, when you expose them at pH 7.8 for about a week or so, and you put them in a Y-shaped olfactometer, they fail to respond to predator odor and swim right in the wrong direction. Now, over the years, this was then associated with the switching on of GABA-A channels by the fish when the fish tried to compensate for the increasing CO2 levels outside the body and rendering GABA-A channels open on a permanent basis. How was that researched? It was researched really by giving it gabazine, which is a compound that is an antagonist and closes the GABA-A channel again. So if the theory is right and you close the GABA-A channel, then they should respond again. And the figure by Nielsen showed that this was the case. Okay, one of the mechanisms in fish seemed to be that the neurotransmitters are altered. But that is fish that has been kept in that water for at least a week. Whilst we saw on the last slide that actually day and night fluctuations in the future are much bigger than the total change we will see in climate change. Yeah, because that's supposed to be 0.4, 0.5, and the difference between night and day in that slide showed you there'll be a lot less than that. So animals are not in a constant environment, so we can't really do experiments where we have them 1 week in one static pH. They experience, especially those many ones that live near coast, they experience pH fluctuations on a daily basis. So these two people here, Clements and Hunt, did a review, and I also had a newer review than this, but 6 years ago, there were already 150 papers from the initial 2 or 3 that were by 2010. And they did a meta-analysis of the behaviors that were changed, and they found really that not all behaviors get worse. About half of them show when you actually acidify the water that animals' responses to the cues are actually going down. But 10% of the cues actually got better. So at lower pH, they responded even better than that. A quarter of the responses, there was no real difference. And in another 15%, some got better, some got worse. The interesting thing is that in many cases, that was actually in the same animal, it's just different behaviors. Let's say the sex pheromone response was down, but the response to food was up. So all that didn't really make sense by explaining it just by one factor, which is neurotransmitter change. So we had a look at that, and we came up with a theory that we made a model of. Here in this one, we'll say, okay, the increase of CO2 and the decrease of pH, which actually go hand in hand and can't really be distinguished well, other than changing the pH with acids. They actually do one thing, they change the cues that are out in the environment. Why is that? Because chemicals, as all chemists will know, do not just occur in one form, but they occur, for example, in differently protonated stages. And the protonation stage can be switched by pH. So chemicals are not just as uniform as people think. They can change, and they can change over pH. They do interfere with receptors, and the receptors are usually peptides, amino acids, and they might actually be pH dependent as well. As a consequence of that one, we'll have changes in receptor odorant affinity and binding. And these then also affect the neurotransmitters and the neural responses that go to the brain. In there is also then now the animal's physiology that has changed by them trying to compensate for high CO2 and activating, for example, GABA-A channels. The totality of that one is altered behaviors. Altered means, in this case, not everything is going worse. Something might get better. Now, this is a lot of theory in there when we started, but obviously we'll have to prove quite a few of these. So what did we do? We started with this one here. And I always put the pictures of the key people who work in that field. So this is Christina Rogers, who's now postdoc, or research fellow actually, did a PhD with me, and Paula Schirmacher, also chemist, doing that. We started with this one. I'll repeat basically the second slide by saying, okay, CO2 into water comes hydrogen, yeah, and more protons means more acidic. More protons actually now protonate molecules, and the protonation of the odor molecules or the chemical signals may lead to these ones having different shapes and protonations. Yeah, and our question then were obviously, this is a chemical law, we need to test it, yeah, and prove this, and then look at what is the effect on the bioactivity. So we did NMR measurements of different chemicals. We produced titration curves. This one is a titration curve of a molecule that is actually a signaling compound, glycine, histidine, lysine, GHK. It's a settlement Q. And this is a dissociation curve. And it basically says that at low pH, at high pH here, we have got the unprotonated form. At the low pH will have the protonated form, and the pKa, in which these two forms are equal, is 8. That's near the pH of seawater, and that tells us already that if we change that, we change the protonation. And here's the place where the protonation changes. So if we now go to the bottom left, we'll see the pKa values calculated, and we'll see over the small area of change between 8.1 and 7.7. Well, we know it's 8.2 to 7.4, so way further left. We'll see that the unprotonated form declines dramatically. The protonated form percentage-wise goes up. And in our example here, between 8.15 and 7.8, it's a change of 25%. Now, if we make this bigger and we have a change in the middle of the night to 7.4, obviously we'll get to this one here. Very far down, we only have about a few percent of the non-protonated form. So then the change is about 80%. Okay, knowing these things, one can actually model that and look at the impacts it has on the structures. That is a relatively complicated process, and David Benoit, physical chemist, helped us with this, and we ran models using our supercomputer Viper, and we had to include, for example, the water molecules surrounding it, but also the salts involved, and all the interaction that has on the molecular dynamics of this one. So it is actually a relatively complicated thing to do, and a lot of computer power is required for that. I don't want to bore you with that, but the net result of that one is that you can make these interesting models here that show you the positive in red and the neutral on the molecule, and that over pH ratios. We then add a bioassay and see what happens. Okay. Oh, am I got one too quick? No. Okay. What sort of systems are we using? Well, initially we started with these ones here. This is the Polygamy Worms I did my own PhD on, really. This is a male here on the left, the red and white one, and the green one here is the female. They reproduce once in their lifetime. The female lays an odor trail of this compound here on the top left. It's a molecule that's a little tetrapeptide. It's glutathione plus cysteine, and then stuck together via disulfide bridge. The female releases significant amounts of these, and the males initially detect that, increase their swim speed dramatically, follow the trail, and once they meet, they swim around each other and shed gametes into the open water. The female dies relatively quickly, or is basically eaten by fish and crustaceans, and the male swims away, but he also dies within a few days. Now, they were so good as research animals because they only respond to sex pheromones, no feeding cues or others. And we could make a clear correlation between how big a female is and how much pheromone she produces. When she swims around, how long she swims determines how much cue she still has. So if a female is out there in the open, she swims and releases pheromone and doesn't attract a male, then after she can't do it another night. Yeah, you can see on the top right here that she's basically out of pheromone. That is because when they swim, they actually make this molecule glutathione into the pheromone, and glutathione has a couple of roles. It is actually the major oxidative stress oxidative stress molecule in the body, and the interplay between glutathione and its disulfide keeps the oxidative status of the cells correct. If you deplete cells, in this case eggs, of glutathione, then the fertilization rate goes down and the larval success is near zero. Okay, it means for them it's pretty important to have these cues working. So if we now come to our theory and say, okay, if we actually have that cue and that cue is pH dependent, what do we get? Well, we get 90% of the pheromone that's released at normal pH, 8.2 at that time, is in this sort of T-shaped form. If we reduce the pH, then almost half of it at pH 7.7 stroke 7.8 becomes this sort of ball, and it's the yellow lines here, hydrogen bridges that appear. Okay, if we lock and key this, that's be the key. The key has changed dramatically. Now we look at that, is we can calculate the amount of active Q. Active is only that T-shaped form. The ball form is inactive, and how much is in the water. And there's a point around pH 7.89 where there's not enough release by the female anymore for the males to respond. That's our calculations out of the thresholds. If we do that in the field, in our experiments, this is exactly what happens. At low pH, the male doesn't detect it, he doesn't start swimming fast, he doesn't catch up with the female, and no mating will happen. So our theory here is actually almost identical with what the behavioral results gave us. Now, the main question we then had was obviously, is it the structure of the Q? Yeah, because we said it's T-shape and now ball form. If we take a Q that is stable around pH 8, then this Q should theoretically, if we drop the pH, still work. Now we tested that, or better Ashley tested that, by using this Q, uric acid, in platinaries, so a relative of the nares, another polychaete worm that looks almost identical, a little bit smaller, and no surprise to us was that the responses were the same, whether they're in low pH or high pH. Yeah, this is a pH stable Q. So totally independent of neurotransmitters or others, if we put them in low pH, they still respond. Whilst, as expected, our peptide Q didn't work anymore. And not only did it not attract them like in the last slide, but also when the concentrations are higher, that didn't release any gametes. So decreased the pH sensitive Q gave us a decreased response. A pH stable Q still worked. Does it have any impact? Does it actually matter? Well, we tested a few other behaviors and we found, for example, that the animals eat less, they consume less, they actually are not attracted to food that much, and all animals in lower pH are smaller. We now do measurements, for example, of metabolic rate to actually underpin all that, but they also produce fewer eggs. So there seemed to be a big impact of that. And the impact study is ongoing right now. This is a new project that we've just started with Victoria Morris, who is an EU fellow joining us. And she's going to use the platinaries simply because they're a molecular model system where one has the genome sequenced, for example, and could look at the molecular basis of all this. So she will look at the production of the Q, how much is released, the transport, whether they change the Q, the reception, and look at receptor interaction, and also the signal transduction pathways. How does she do that? Well, she made this slightly complex looking slide, but it's basically the one on the left is just showing that all sorts of animals don't behave normally anymore when we drop the pH, but on the right it shows you that what she will do is she will test the behavioral responses, she will test the production by measuring the Q, but then she will also do transcriptomics, deorphanized receptors, because we know the receptors roughly where they are, and then we will make knockout candidate genes, and then eventually we'll make worms that do not have that chemoreceptor, and they should theoretically then behave very differently. This is all done with in collaboration with Gaspar Cecchini in Exeter, with his help there. So we're looking at the real impacts on the receptor. We also have another project together with Southampton University and CFAS, and that looks at a slightly different aspect. It's again looking at narrate polychaetes, but this one is really looking a little bit more into the wider picture of this. So in an ecosystem where you do have animals like our polychaete worms, if we have functional traits and they're affected, is this something that over generations the animals find alternative ways? So can they adapt to it? Can we see this? Is this really a molecular change? So we do genome sequencing. We've just finished the nares divesicular or histidine divesicular genome. We're doing transcriptomics and then gene methylation and whole genome sequencing to see where there are small changes. And we'll hope that we'll have more generations. So we're on F6 generation. We already see that the animals in there are responding much less to food, and they are way more relying on visual cues, and they're coming out a lot more during the day than they normally did. The molecular work is still ongoing. This is part of the biggest bigger study with more animals and more species. And eventually the idea is to make a trait-based predictive model to look at ecosystem responses. Okay, what else do we do? Well, we work not just with worms, but also with crabs and crustaceans. So this is the other species group that we look at. Here is shore crabs, Carsanus minas, that form pairs for mating, and the pair formation happens due to pheromones that are involved. Equally, the pair formation in these Lismata cleaner shrimps, that one is done by a little compound called like octadecane amide, so it's an amide in here, whilst this one, eagle eye, people will see that's a simple nucleotide that comes and is released in large quantities during the molt and the chitin biosynthesis. These are actually also the animals that we look at for pest control using those cues. If we expose shore crabs, but also the cleaner shrimps or arrow crabs to low pH and give them the pheromones, they really have a much reduced response. John Tershek here found really that if you look at the protonation stage of the molecules, this UDP, it has a ketoenol tautomers, and they'll change, and only one of them, I think it's the enol form, is bioactive, and I think it's the enol form, yes, and the keto form not active, and in lower page, it all switches to the enol form. We then also looked at feeding compounds in those. We used mud crabs to do that. And we used known cues like betaine, that is pH stable, respond in low pH and high pH is the same, no real difference. Glycine, that is slightly affected by pH, has a lower response. And muscle homogenate, which is obviously lots of chemicals, mainly amino acids, has a lower response. And the whole thing does happen immediately, or also if we have them in the cultures for about 4 weeks. We're currently redoing these experiments with fluctuating pH. This is the first study that we published to show how the molecules are affected. This is Christina's work on the shore crabs and many other crustaceans that carry eggs under the abdomen, and the young as well. And they pump fresh water over the young, and before they actually hatch out, the young release a lot of this compound in order for the female to pump the eggs away. So here's the 3 chemicals that are involved, and we have, for your convenience, circulated here the protonatable groups on them, and these 3, the 2 tripeptides and the dipeptide here are all involved. Christina made the dissociation constant measurements and the prediction of the protonation stage, and on the right you'll see what happens with the 3 cues, and you don't need to be a chemist to see that from non-protonated green they go to protonated red, and their form and shape is changing as well. And again, it's no surprise then really that the bioactivity of these cues went down. Now, interestingly, these cues are not just used by shore crabs for hatching of the young, but it's also the same chemical, GHK it is, this one on the left here, that induces the shell-seeking behavior in hermit crabs. So it has a lot more impact, and the larval settlement in oysters and in mussels is also induced by these peptides. So widespread. But not always did we find a major response in terms of less bioactivity and a changed behavior in molecule, and could attribute that to the changes in the molecule. Here we have a compound phenylethylamine, and to the right you will see the protonated form here in red, and you'll see that the protonation changes, the structure not that much, but the geometry changes a little bit. Interestingly, around pH 7-8, around the ocean acidification relevant pHs, the changes are minimal. Still, we find a significant change in behavior. How could that be? Well, we had a look at it, and the next slide, if you look at the molecules here, you'll see the molecule PEA on the left and the protonated one on the right, how they fit into the receptor. This is a TAAR1 receptor that we modeled, and we modeled the interactions of the molecule and how it fits into the active site of the receptor. And you probably have to watch this video once or twice to see that actually the molecule doesn't fit in properly anymore and it sticks out a bit. So the protonation stage now actually affects the receptor process. The next step, what we want to do is actually to look at the active site and how that one changes. We do work in collaboration with the University of the Algarve, where I've been for many, many years. And we did the same thing with feeding cues. Here's your vertebrates, some fish here, sea bream it was, gilt-head sea bream, and it showed really that the animals can smell the compounds, but they need a lot more of it. Yeah, remember the bioactivity or the bioavailability of the correct form goes down. So here's the correct form, here's the protonated form, but obviously not 100% of all molecules released are in the protonated form. And you can see that I need a lot more. We did find some weird things when in the sea bream as well, which was that around the nose, they had an increased amount of mucus, presumably as a sort of response to the decreasing pH. So we concluded that really our initial theory that the chemicals change and therefore reception and the receptor ligand interactions change. And we made this slide here on the left here to show that on the worms. Mr. Wert published a few years ago a paper showing that this change of olfactory cues in the water is one of the 3 main problems that animals face in changing oceans. But we didn't stop there. We wanted to know a little bit more. We wanted to actually make sure that we can predict this. We know we can predict the responses of the animals, fine. Yeah, we can see that one pH or the other, they do respond or not. We can predict and model the chemicals. We can actually, and increasingly can do that, model the receptors. But we can also hopefully, hopefully make geospatial models. And this is where things are also new from last time around. So here is just a predicted pH and pH change on a global scale in the oceans. But if we actually look at the bioavailability of the active form of a molecule, we can predict that. So what did we do? We had a look at these 2 compounds. One is tetrodotoxin, also known as pufferfish poison, and used by some of the pufferfish as sex pheromone. And the other one is saxitoxin. It comes from harmful algal blooms, HAB. And the protonated form of this one is the toxic one. So again, what did we do? We looked at the protonation stages, how the molecule changes, did our NMR analysis and the pKa, and we find that the more toxic form increases dramatically when you drop the pH. Fine. If we now overlay that with the places where harmful algal blooms actually do occur, this is the black dots here, then you can actually go and look at the predicted pH in those areas, and that changes from the non-protonated to the protonated form. So if you have the same number of molecules, let's say I've got 100 molecules, you will, in the water, you will then have 30% more of these ones in the active, in brackets, more toxic form. If you do these calculations, you'll find out that Australia is one of the places where the changes are dramatic. And you'll also see that where we are here in the North Sea, there is quite significant change. And here where all the fish grounds are off the coast of Russia. So one can do these changes, one can do these predictive models with all sorts of molecules. And one can then try and link this to effects. And we did this by linking up with tribes in Canada, Indian tribes, and they found that, and we looked at the effects of the saxitoxin and the increase in butter clams. Here's the species Saxidomus gigantea in the tissues and how the toxicity would change. And interestingly, for the last 7 years, the toxicity was so high that it exceeded FDA limits, and it has obviously effects on the predators that eat the clams, seals, for example. So now with the models changing and making the predictions even worse for pH drops, we probably have to adapt this one, and we'll probably see even worse figures here. You can also use that to predict something totally unexpected. Here is a paper that we've just submitted. We put it on bioarchives for a short while, and it is, we actually looked at tetrodotoxin in amphibians. And this is with Katharina Wallenberg, who is a specialist on this. And we did the same thing. We made global maps. And the interesting thing that we found out was that the most toxic of these frogs, for example, that have high levels of tetrodotoxin live always in those areas where the pH of the soil or the water is very low. There's a clear correlation between the number of species in those ones and the toxicity increase that comes from tetrodotoxin being more toxic. What does it mean? It means that really we know now that increasing CO2 will have an impact upon the olfactory system of the animals. It will possibly make them release fewer compounds because they're stressed, i.e. by neurotransmitter changes. We'll see that the cues once in the water change. Once in the air, we don't know that, but as the chemoreceptors are not dry, but there's always a liquid interface, we expect there will be something to happen there. We know that there's impacts of temperature also. And we know that seasonality plays a role because in the summer, the pH is different to the winter. For example, in water, CO2 dissolves much better in cold water than it is in warmer waters. So what is it in vertebrates? Well, here's a few pictures of mice, for example, and here's your lovely lizard. I do scent marking. Does it mean when it's drier or with a higher CO2, the molecules dry out quicker, the volatility half-life changes? Does it mean the cues change when they fit onto a signal receptor? Does it mean in very high CO2 levels, we see something like we see in fish, that neurotransmitters are affected? Do we see receptor ligand interactions? Does it change the animal physiology and the fitness? Does it therefore have impacts on food availability, different behaviors, social hierarchies, ecological networks? The answer is maybe. Very little is known. And I think there is a huge potential for research on vertebrates on this one, including man, by the way. And I'll have, I don't know whether I have much time left, but I'll try to get to this one here. This is odour molecules that are released from plankton, dimethylsulfane. It is known that dimethylsulfane will probably go up because we've got more plankton, more phytoplankton in the warmer waters, and more CO2, and it affects seabirds because they detect this as food. And down here in blue, I put something in that you might know already, because plastic in the ocean is nothing other than a big surface area for biofilms, and when biofilms grow, they release dimethylsulfane and DMSP, and that attracts seabirds. There's a few papers on that one, also some on fish. So what did we do? We did the same thing. And here's a little study that Jack Greenshields did with us. We looked at the reasons why the plastic may have an odor. And we found this compound oleamide, that is a slipping agent, is released. And you can see out of the video that if you give this a little hermit crab, the hermit crab really is excited and runs to that oleamide and is attracted to that. So it's not just fish or that are attracted to it, or seabirds, but marine invertebrates. And in this case, we actually know really what Q it is, because this oleamide here is a known chemical signal. It may well be a slipping agent. A slipping agent means that the water drops off better, but it is also known from Dongzhang's studies 10 years ago that it induces the mating behavior in the smarter shrimps, in those cleaner shrimps here. So it makes the hermit crabs believe that the plastic odor or the oleamide has something to do with food, and it has in the cleaner shrimps potentially the role of inducing their mating behavior. Why is this important for climate change? Well, here we go. Our latest data show that if we decrease the pH, it becomes even more attractive. And it increases the attractiveness, for example, of polyethylene for crabs. So this may well be because they can't smell the other odors that well. If we give an animal food on one side and polyethylene on the other, and we do that in low pH, it actually chooses the polyethylene over the food. Right, last little topic. How do we explain all that to school children or people who are non-experts? First thing I said, we make a cartoon. Sounds great. So one of our ex-students, Grace Marner, her name is down here, she actually visited our lab when she was doing a master's in Sheffield, and said, "Jorg, can I work with you?" And we made these little cartoons here to show to children what the differences is, that you locate food by odor, that you find mates by odor, avoid predators by odor, and what molecules they are. This started a major program on interactions with schools and reach out. And here is a little video that I don't play now, but if you have the link, go on that and look at it. It's about 8 minutes long. And it explains to school children and the public really how oceanification works and why odor cues are not working anymore. It's a really cute animated one. Grace actually won a Royal Television Award for that 2 years ago for the best student work. She presents that nicely here. And it was really all part of that package aimed at CBBC audience, children from 6 to 12. We took this further, and Helga and Christina went to schools and did a school project, and you can see the kids doing experiments. Yeah, here's a class of school children. We gave the hermit crabs. I love this picture when the hermit crabs in this glass shell that our glass blower made. You can really see how they look like. Wonderful. And they did experiments, very simple ones, to see whether the crabs run to food or not. And as no surprise, surprise, at normal pH they do, and it's very fast. At lower pH, fewer do that, and if they do that, they take longer. And you can see that the results were very similar in school classes as well as our science festival. And we've redone the whole thing with scientists, and they came up with exactly the same data. So it shows that this is authentic science, and the data are really trustworthy because they come exactly the same data. These children didn't have a clue about that beforehand, and naively just observed and said, "Miss, is that?" And they come up with exactly the same data as our highly trained postdocs do. So it's real science. They published this in a journal, and we asked, or the teacher also asked the school children then to put this into a poster, and we've seen some beautiful ones. Yeah. So these are kids that are 10, 11 years old, and then it made these wonderful posters. Here's a link if you want to see the publication. And Christina then put this a little bit further. She went to COP26 to show off the game that we made. It's called Krabby's Reef, and it is a visual audio cues where you follow a maze to find the exit, and an increasing fork is there to disturb your vision to simulate what happens when ocean acidification happens. So here's the links for those ones if you're interested in that. Here's the whole lot of people that were involved in that. Obviously, this is quite a significant amount of people, and not everyone is here on the photo. But down here is the current members of the research group momentarily. We are actually 21 in that research team. So it is a significant size of group, but by the diversity of the work that you've shown you probably appreciate that. And that's my presentation for today. So thank you very much. And I'll be happy to answer questions.
Thank you very much indeed for that absolutely fascinating, wide-ranging, important and frightening talk. Really very enjoyable.
I'll just push the light on, otherwise I'll sit here in the dark.
Okay.
Hold on, here we go. Yes.
Okay.
Better. Yeah.
I love the bit at the end about teaching children. I think, as we talked at the beginning, it is hugely important to get that generation involved and restoring the planet to how it ought to be. I'm just going to look for questions. I've got one. I was very surprised to learn what the ocean pH is, because of course we have pH around 7.4 and it can only change by about 0.5. And yet we at some point came out of the ocean.
Yeah, that's true.
Yeah.
If you think about it, I guess you look at a long scale evolution there, because the pH around 8.1, 8.2 with 300 to 400 ppm CO2, that's been stable for what, 35 million years. So I guess all communication systems have adapted to that as being optimum.
Yeah.
And it makes sense that the whole thing is down to the chemicals and their structures, because they only have to optimize on the protonation stage that is the most abundant at that pH. Yeah. So we weren't surprised that most of the cues that are identified in the aquatic environment are pretty pH responsive, and lower pH will change their structures. It doesn't mean that in the future they might not be able to adapt. The worry is only that it's so quick. Yeah. And the other worry is that the extremes are so bad now. Yeah, the average ocean pH going down by 0.4 is bad enough, but it's when it goes down to 7 or well below that, then it has an impact. I haven't shown the link to the video of the upwellings on the coast of Oregon, for example, where a lot of behaviors are not happening with crustaceans in the summer. So in the main summer, it's warm enough, the water, but the pH is well below 7.5. There's no mating in shore crabs there, for example. As soon as the pH is higher up again, they respond again.
Wow. Wow. Krishna has a question for you. Would you like to ask it yourself, Krishna?
Yes. Thanks, Jorg, for an excellent talk. Yeah, I was just wondering, we've got several problems associated with pH changes in the environment. Mm-hmm. One is, if you have lots of amines that are cues in the environment, obviously the protonation is dependent on pH. Yeah. But not just the pH of the amine itself. What about the animal itself? Because obviously the animal is full of proteins and amino acids. Obviously the conformation of receptors will change. Mm-hmm. Do they just get sick and they can't detect the cues?
Yeah, I think there's two things here. Short term, probably not, because the short term is, if you think about I work with shore crabs. So shore crabs right on the coast and on the Algarve, they'll get pH changed twice a day. So it goes up and down, up and down. It's a bit like visual cues. Yeah, during the night, we're pretty useless. During the day, we're fine. So animals will probably just wait it out. Yeah. So when the conditions are good, they'll forage. When the conditions are bad, they just wait. The waiting times might be longer, especially if you're a night hunter. When the animals are in a more stable environment and the pH drops and stays low, then it is a little bit sort of getting sick, because unless the animal can compensate for the high CO2 on the outside, then the CO2 level, and with it the pH in the body will go down. That happens to, for example, crustaceans. And a lot of the body functions will differ, and you're absolutely right, because the next thing that will change is hormones in the body. So lots and lots of these things will change, but they haven't really been studied well. Yeah, so we have to be really careful of not mixing the two short-term and long-term. And the research so far was a little bit one-sided, because everybody kept them in one pH, 8.1 as control and 7.7 as treatment. Yeah, and stable. And there was massive amounts of discussion to keep it within 0.001 of pH. When in reality, it fluctuates daily, winter, summer fluctuations, etc. And one of the adaptations might just be that the animals just wait it out until the conditions are better.
Okay, yeah, thank you. Just one more question, please, Jorg. On the model that you showed of the docking of the glutathione compound with the receptor, did you also look at the effect of pH on the receptor itself?
No, this is, as I said, what the next thing is going to happen.
Yeah.
One of the projects that we have, for example, that EU project, it really is to characterize the receptor. And we tried to work with a species that we normally work with, an Aries succina, but they're very difficult to breed and molecular-wise not much known. So we now tried its relative, these platanoids, where the genome is known, and we know all genes, which receptors are encoded, etc. So if we then tested whether they respond to the same cue, which they do, so we're now in a situation where we can now use advanced molecular tools and to have a go at the receptor. Once we know which is the active receptor, we can model the active receptor, the part of the receptor. The little bit was done on this PEA. That was a TAR1 receptor where the structure is known and a 3D structure as well. And Paula, who did the work, she basically in the model cut out the active site plus a few amino acids in each direction, and modeled the receptor itself, where it showed that they fit in and wobble in differently. Yeah, that video. There we did model the receptor already, yes. But we'll do a lot more on that in the future. It's quite challenging, really.
Okay, thank you.
Yeah, the computer power that you need for that is significant.
Yeah.
We don't want to contribute to climate change too much.
Is there, Jorg, any link between temperature change and acidification? Do they go more or less in parallel?
No, in opposite actually. So CO2 dissolves extremely well in water. And it's much easier to get it in water than out. This is one of the problems we have, but it dissolves more in cold water. That's why, for example, these upwellings on the coast or on the west coast of the US are having such a low pH, because more CO2 dissolves in it. So a little bit saving grace for us is that the CO2 is not as well soluble in warmer water, but it's nowhere near enough to compensate for the drop, I'm afraid.
Okay. Shame.
Yeah, I thought that'd be great, wouldn't it?
Yes, yes. And I think you should show Scott Morrison that lovely map you've got with Australia, the great brown mass down the side of Australia.
I think this is one of the most potentially most interesting parts of the research, because as I've seen with the example of the amphibians, yeah, which was quite surprising, because we looked at how toxic the compound is, on different pHs. That was known, and we applied that, and Katarina found really that all the frogs that are known to have high amounts of toxicity are living in soils with low pH and waters in low pH. So they've probably evolved in there being better protected. Yeah. But one can do these models with everything really. And yeah, unfortunately, I never learned GIS. If I'd be a student now, I would learn it, because it's very important to make.
Yeah, absolutely.
So we can actually basically tell tipping points. You give us any molecule, and we can tell you where on the planet and when it will change a different form. Same thing holds true for whatever toxicity of heavy metals. They are pH
Right.
dependent, so all that can be modeled and on a global scale and in a local scale.
Gosh.
So it's a really, really
Yes.
interesting tool. That's why Nature Climate Change took that paper, really, because of the modeling potential it has.
Yes. That's amazing. Do we have any more questions? Let me see. No? Nobody else? Well, I think it remains for me to thank you very much indeed. It was absolutely worth waiting for, Jorg.
Oh,
Real pleasure to hear you. Such great
Yeah.
science. Thank you.
I love coming to these events, so thank you very
We
much on my side.
very much like having you. So do come again. Thank you very much. We do have another meeting in December, and it's going to be Jenny Tillotson talking about her work with releasable fragrances and a possible anti-COVID use as well.
Cool.
So I hope that will be of interest to all of you. And it just remains to me to say thank you again to you, Jorg, and to Simon O'Connor, who organised this, and to Krishna, who kindly volunteered to stand in for me if I didn't get back in time, but I made it. And we hope to see you all next month.
Okay.
Bye, everyone.
See you. Bye.
Bye.
Thanks for having me.